Hey everyone, so we're sort of getting into the nitty gritty here, the last month of class, which is just crazy. I can't believe how fast everything has gone. Um, and we've sort of been all over the place due to circumstances out of our control for these past few weeks. So what I'm trying to do with today's quick lecture here, sort of bring us back together and get us on track so we can get into the lab and knock out what we can knock out while we're still in lab together. So today's lecture is going to focus primarily on mutations, and that has to do with changes in DNA bases that ultimately will impact protein synthesis, right? Or may not, but those are the processes that we're looking at. And you've been investigating those really over the past couple of weeks. So what I'd like to point out here to you is just a few things. Here we're looking at DNA, and DNA is shown in that dark green band here in the back, right? And you're going to see this image quite a few times as I go throughout these notes today. And then what we're saying is, like, let's look at just this section right here, which has all these, sort of fancy regions on it. And I want to explain some of those fancy regions and what they mean. Let's get a closer look. So we're zooming in right here. And we're going to look at some different parts of DNA that aren't just coding parts or what we would call genes, right? So here in purple, what this represents is a piece of DNA that codes for a gene, right? There's going to be a start codon, there's going to be a stop codon once the RNA is made, and a protein is going to be made. But before that, there's these really special regions on DNA that we haven't talked about yet. One is called a promoter, and that's shown here in yellow. And the way that we can think about a promoter is almost like a dial or a switch that can really make a lot of mRNA or can make a little bit of mRNA. And then there are these couple of regions back here, which we'll talk about, which are shown in blue, called enhancers. And enhancers are sort of like they're time dependent and also location dependent. So an enhancer might be switched on in one particular cell, but not in another, depending on how much of this gene actually needs to be expressed. So what we want to talk about is how can mutations occur in each one of these regions and what's the impact of those going to be. And we've already covered some in the coding part. And that's what we'll discuss today. So today should almost be a little bit like a review. What we talked about last week with gene level mutations is that these are point mutations, right? And then in those point mutations, we talked about a substitution of a single nucleotide, one single nucleotide, or we could also have the insertion or deletion of some nucleotides in these regions, right? So we talked about those. There's also these other types of mutations that could cause repeats. So there could be multiple repeats within a given section. I'm primarily just going to focus on the point mutations and the frame shift mutations with you guys. In chromosomal mutations, right? So these are different from the gene level mutations. These are going to impact entire chromosomes. We can see duplications, deletions, and rearrangements of large sections of DNA. So we could have like this entire region circled here in orange. We could actually have that duplicated on a chromosome, right? Or maybe that region right here could actually get entirely deleted. Those are examples of chromosomal mutations, right? Which have a large impact on the organism, right? So all of these are going to have large impacts on organisms. This is an important note to make. As mutations produce genes with different sequences, right? So whether that's in the coding part or it's in different regions, each different form of a gene is called an allele. And we've used this word before when we're talking about disorders, right? And we're looking at things like homozygous dominant, homozygous recessive, heterozygous. Whenever we have these mutations, this is what causes these different forms of genes to happen, or these alleles. It's through mutation, right? So we're just looking at the molecular basis of alleles now, and that is through these things called mutations. Here are some causes for point mutations that I wanted to go through with you. Like, how do they actually pop up? Well, one way is spontaneously, right? So this could be primarily during eras of DNA replication, which we will talk about in class this week. We're going to touch on replication a little bit, but really we're going to steal this idea of replication for an application in the lab. Another could be free radicals, which can alter nucleotides. Another could be mutagens, like ethidium bromide. I told you guys was a mutagen, right? So these could be chemical agents or radiation. The idea is that these strands are going to separate at some point. and then we're going to add DNA bases back to them. And that's the idea of DNA replication, right? Because we need to make copies of DNA when we're making new cells, so each cell has a copy of that DNA, so it can make the proteins that it needs to. Now here, let me rewind that for you. You can see this is double-stranded, and when DNA needs to replicate, it's going to separate, and then another enzyme is going to come along and start adding the complementary bases. And what can happen is a mistake like right up here, right? So we can get a G inserted up there instead of having an A. And that's an example of a point mutation that could happen. One way these can happen is through exposure to chemicals, right? So here you guys can see this is the base adenine. It likes to pair up with thymine, right? Like we know that. But if it gets exposed to certain chemicals, there's chemical changes that can occur to this base, specifically in this region that's highlighted here in red. And if this change happens to occur, it will now pair with cytosine, this base, when instead it should have paired with thymine. Right? So like there's these chemical mutagens that can cause chemical changes to happen in bases, which can then cause them to mispair, and that's a mutation. We can also, through things like, ultraviolet light, like light that comes from the sun, which is very high energy. It can actually hit and impact our DNA because those wavelengths are so small of that light. They actually can bump into our DNA, and they can cause these things like thymine dimers, where these thymines will actually stick together on DNA instead of being bonded to their adenine bases like they should be. So there's lots of environmental cues that can happen, but there's also these spontaneous things that just happen accidentally in the cell. Now, here's what we want to get done with in these notes. I'm not going to do all of this today. What I'm going to focus on today is just part one of this. So what we want to look at is the effect that mutations have overall on the organism or the protein really being made. That's what we want to focus on. Today I'm going to focus on what part of the gene is actually mutated, right? And then we'll talk about the function of the affected gene and then the timing and place or the cell type of the mutation. That's what we'll go through today, is this part one. So here's our image again. Remember, this is a blown up image of what we had on that previous slide, where we've got the enhancers and promoters and the coding part of a gene. These are the different regions that we can look at, right? So we can look at the coding part of a gene, this is going to tell us about the function of the gene product, or what we would call the protein function, right? Because we know a protein is going to get made ultimately from this DNA. The effect that we're going to get here ultimately depends on the amino acid sequence, and then how those amino acids are going to fold up and make a protein. So that's what we're looking at in the coding region. In the enhancer region, it's about time. So like, when in development is this gene supposed to work, right? And then where, the place, and then also the amount of the gene product. So if there's mutations in these enhancer regions, these are the things that can be impacted. And we'll look at those more closely in a later lecture. Promoters are going to be basically like dimmer switches on lights, right? They're going to give us an amount of gene product depending on the promoter that's there. So what we want to focus on today, again, is we just want to focus on the coding region. So we'll talk about the enhancers and the promoters. I'll talk about those later, but today I just want to sort of review what happens in the coding region, right? So let's say that we've got this strand of DNA. If you guys would for me, as you're watching this, you should already have some paper and be taking some notes. If not, you need to go ahead and grab some right now, because what I'm going to have you do is some stuff with this strand. And let's just practice a little bit. So I've got this DNA strand that's written right down here. My question for you is, what mRNA and amino acid sequence will result from the expression of the gene below? If you feel really comfortable, you could just go ahead and do this now, right? And sort of pause the video and see if you can get the amino acid sequence. If not, you should stick along with me. So go ahead and start getting the mRNA sequence and the amino acid sequence if you're comfortable. Just pause me right here. I'll be back and review this for you when you click play. If not, you need to watch and you need to take some notes here, right? So I want you to note that the sequence below is written in 3' to 5', that direction, okay? So this is the 3' end, this is the 5' end. The first thing that we're going to do is transcribe the DNA, all of it. And I'm going to show you right down here that if we're transcribing, we're going to make some mRNA. Right, so here's my mRNA copy. And basically what I'm doing is I'm finding the complementary DNA sequence, or really what I'm just doing is pairing up my mRNA with what this sequence is reading here. Right, so A pairs with U, T pairs with A, and you guys are getting the sequence that's written down here. Number 2, I want to find the first start codon, because now we're going to go ahead and translate. And pretend you're a ribosome and you're going to go ahead and start reading this mRNA. I want to find my start codon. Okay, well there's TAC in the DNA, which means AUG is right here in my mRNA. So that's where we're actually going to start. We're not actually going to read these bases. Next, we're going to set the reading frame, and the way that I did that in practice with you guys is I just underlined the codons after that. So let's do something like that. All right, so we can start reading in triplets, and this makes it a little bit easier. Now I want to translate each codon, right? And I want to put its appropriate amino acid. So I'm going to need my chart in order to do that. I need my codon chart. I gave you guys one last week, right? Or you can just look it up on the internet if you lost it. So AUG is my first one, and then I'm going to start plugging in all these amino acids until I get to my first stop codon, which is not an amino acid, right? It's actually just a place that says stop. So now I've got my sequence, and this is going to fold up and make my protein. That whole process is called protein synthesis and how we express genes. Okay, so now that we've got that, we can look at some more specific examples of our mutation. So here's our original strand. This is what we just did, right, on this previous page. This is exactly what we did. Now a substitution is when we're going to actually alter one nucleotide, and then probably that will give us a change in one single amino acid. So here's our DNA, right, and what I'm showing you here is instead of having TACATG, like we read up here, we're just going to replace an A. That red letter A is going to be a mutation, right? And it's just going to be a single substitution, one A instead of the G up here. Now let's see what happens. What's the effect, right? So here again is my start codon. This is going to change our mRNA to a U instead. And if we actually go through and we translate all those, we get the exact same amino acid sequence. So if we have the same amino acid sequence, what do we call that? Well, we call that a silent mutation. It does not alter the amino acid sequence, so it's silent. And this is because of the redundancy in the genetic code, right? That this third base, it will actually code for the same amino acid. So we don't even have to worry about the mutation, right? It's not a problem. Now, let's say that we have a slightly different change here, right? So we've got this C is changed. So we read CTG instead of this one up here. So we've got another single change, right? Which means I'm going to change this to a GAC instead of UAU, right? So we've got this change. Again, this is just a single substitution. Now, if I look at this, I can see that this asparagine is made instead of the tyrosine. So my amino acid sequence did change once I translate it. That's an example of what we call a missense mutation because it's going to alter ultimately one amino acid. Right? So that's a missense mutation. Now another one is what if we say we have TACATT right here. So again, it's one single substitution, right? We have a point mutation that's occurring here. Here's my mRNA, and here's my protein. And now what changed here is that we had a stop codon prematurely put into the protein, which can be disastrous for the function of the protein. And if this occurs, we call this a nonsense mutation because it's going to create a premature stop codon. Okay, so those are my three when it comes to point mutations. When one nucleotide is going to impact an amino acid. If it doesn't, we call it a silent mutation because there's a redundancy in the genetic code. If it does change an amino acid, it's called a missense. And if it puts in a premature stop codon, it's called a nonsense. Okay, so that should be review from last week from some of the things that we did. We're going to stop this video here, and we're going to continue, please continue to part 2 of this lecture in the separate link.